Hello there, boys and girls. Hopefully, all of you are well and keeping safe. So, given the current circumstances, the college has taken a great initiative to utilize the online platform so that you do not lag behind and can complete your lessons in time. So, today we're going to discuss about chapter 3, which is cell division. You know that the body of every living organism is made up of a cell, which is the structural and functional unit of life. The life of every organism starts with a cell, and every cell is originated from a pre-existing on by the process of cell division. Cell division is the process by which a parent cell or a mother cell divides to form two or more daughter cells. Cell division is a very natural and important process occurring in every living being, be it an unicellular or a multicellular organism. In case of unicellular organism, cell division takes place for the purpose of multiplication. For example, in the body of amoeba or in the body of bacteria. Then, on the other hand, multicellular organisms, the cells of multicellular organisms undergo division so that it can attain its multicellular form. You know that in multicellular organism, during a sexual reproduction, two different gametes fuse together to form a unicellular zygote. That unicellular zygote then undergoes consecutive division and gives the multicellular organism its multicellular form. Types of cell division. Cell division can be of three types. These are amitosis cell division, mitosis cell division, and meiosis cell division. Amitosis cell division is the characteristic of unicellular and lower organism where you can see that the nucleus of the cell first starts to elongate and constrict in the middle. In the meantime, the cytoplasm and the plasma membrane of the cell also starts to constrict in the middle and thus form a furrow. With the progressment of the division, the furrow goes deeper and the nucleus also constrict more and thus directly forms two daughter cells. Then mitosis cell division, which is also known as equational division. It usually takes place in somatic cells. In case of mitosis cell division, the nucleus and the chromosomes both divide once and thus form two identical daughter cells. Identical in terms of that the number of chromosomes, the physical and structural features of the chromosomes of the newly formed daughter cells will be exactly similar to that of their mother cell. Mitosis cell division helps in the growth and development of living organism. Later, now we are going to discuss about the stages of mitosis cell division. Mitosis cell division is a combination of two consecutive processes. First on is karyokinesis, which is the division of the nucleus, followed by cytokinesis, which is the division of the cytoplasm. Now, cell division is a very complex and lengthy process, and therefore to undergo division a cell must prepare itself. This preparation phase is known as interphase. In this phase, the cell various changes occur in the cell that makes it compatible for undergoing division. Mitosis cell division, for your better understanding, can be divided into 5 stages. These stages are prophase, prometaphase, metaphase, anaphase, and telophase. So the first stage of mitosis cell division is prophase, where in the beginning the nucleus of the cell becomes larger in size, as well as the chromosome of the cell starts dehydrating and condense, giving them a shorter and thicker form. So this was our mother cell. That means this is the condition before cell division. During division, this is the first stage of prophase, where you can see that the chromosomes are shorter and thicker and more visible. At the last stage of prophase, we can see that the chromosomes, except for the centromere, divides to form two chromatids. These chromatids are called sister chromatids. In case of animal cell, the centrosome divides and forms centrioles. These centrioles spreads aster ray. Animal cell contains centrosome. It is the feature of animal cell. In plant cell, centrosome is absent. The second stage of mitosis cell division is prometaphase, where in the beginning a spindle apparatus containing this, you can see that this is the apparatus. The spindle apparatus containing two distinct poles are formed. The middle plane of the spindle apparatus, the middle portion, this place is known as equator. Now you can see that in between these two poles there are some fibers that are stretching from one pole to another pole. These fibers are actually made up of microtubules of cytoskeleton. Later, the chromosomes starts to get attached with these fibers. They actually get attached in the centromeric region. That means where the centromere is present. A figure will explain it better. So this is the figure of a chromosome during a division. So these are the two sister chromatids. This is the centromeric region. Here in the centromeric region there is a protein fiber called kinetochore. This is where the mitotic spindle microtubules, that means the spindle fibers, gets attached with the chromosome. The fibers that gets attached with the chromosome are called traction fiber. These are also called chromosomal fibers, that chromosomes are attached with them. Chromosomes then start assembling on the equatorial plane. The chromosomes that were towards the pole, they starts to come near the equatorial region. The nuclear membrane and the nucleolus starts to disappear and disintegrate. You can see here it's not prominent like before. And in case of animal cells, the spindle apparatus is formed by the centrioles. Here the centrioles, they remain towards the pole, and astral rays, you can see these are the rays, astral rays are spreading from them. So the third stage of mitosis cell division is metaphase. So at the onset of the stage, all the chromosomes assemble at the equator. So in the prometaphase, the chromosomes started to gather here, and you can see that in metaphase all of them are in the equatorial region. None of them are near the pole or anywhere else. So all of them are here, and the centromere, you can see the centromere of each chromosome remains on the equator, and the two arms take positions towards the poles. Metaphase is the stage when the chromosomes becomes the shortest and thickens and make them most visible during division. Fun fact is that this is the only stage where you can actually count the total number of chromosomes present in a certain cell. In addition to that, the attraction between these chromatids, these two sister chromatids, they starts to decline. This attraction declines and they start to repulse each other. Okay, so at the end of this stage, the division of the centromere starts. As the chromatids starts to repulse each other, the centromere starts to breakdown. Then you can see that nuclear membrane and nucleolus has completely disappeared in metaphase. So if the question comes, in which stage of mitosis cell division does the centromere and do the nuclear membrane and nucleolus disappear, the answer would be metaphase. The fourth stage of mitosis cell division is anaphase. Here in the metaphase, the centromere started to break down, and in the anaphase you can see that the centromere has divided completely, making the two sister chromatids sister chromosomes. They have become individual chromosomes, you can see, and they have started to move towards respective poles. In this sort of movement of the chromosome, you can see that the centromere is positioned towards the pole, whereas the arms are positioned towards the opposite direction. As each chromosome splitting into two moves on the two poles, as a result, the number of chromosomes remain unchanged in the newly formed chromosomes. So in anaphase, on the basis of the position of the centromere, 4 different types of chromosomes can be seen. This picture explains it better. Here you can see when the chromosome is present in the middle and the two arms are almost similar in size. This takes the form of V shape. Then it is known as metacentric. Then it looks like an L because of the position of the centromere towards one arm. This is known as submetacentric. Then this is the J-shaped chromosome and known as acrocentric. The last one, when the centromere is present nearly towards the corner of one arm, this is known as telocentric. At the end of the anaphase stage, the daughter chromosomes are completely pulled apart. They are completely pulled apart and they take position to the poles and they start to elongate. And that's when anaphase stage finishes. When the sister chromosomes reaches the respective poles, the anaphase stage finishes and telophase, which is the last stage, starts. It is basically the reversal of the prophase, which was the first stage. In this stage, the chromosomes starts to decondense. They get hydrated and then get surrounded by a new nuclei. These chromosomes starts to intake water, and as a result, they elongate and become thin and long structures, and these are then termed as chromatin fibers. These chromatin fibers then group together and form a mesh of chromatins known as nuclear reticulum. You can see here that the nucleolus also reappears, and as well as the nuclear membrane surrounding this mesh of chromosome, that is the nuclear reticulum, also reappears. And as a result, you can see that in two distinct poles, two different nucleus are formed. The spindle apparatus disintegrate, and as a result, the spindle fibers gradually disappear. At the end of telophase stage, some small purse, these purse, it actually comes from the endoplasmic reticulum. The cell contains endoplasmic reticulums, and from that reticulum, some small parts, they aggregate near the equator. This is the middle plane, the equator, and collectively they form a cell plate. The cell plate is where the cell will divide and will form two distinct daughter cells. Here you can see that, as well as in the equal distribution of cytoplasmic organelle. The mother cell equally distribute the organelles and they go to respective daughter cells, and thus the cell plate is where the cells gets divided and forms two daughter cells. In case of animal cell, there is no need of cell plate because cell wall does not form in animal cell. So as a result, the plasma membrane forms contractile ring. It forms a ring. It just goes inside and forms a ring, and then these two rings pinches off. They get together and then divide to form two daughter cells. So this is how mitosis cell division takes place in living organisms. So why mitosis cell division is so important? Mitosis cell division is important because it plays immense role in the growth and development of multicellular organisms. Because if mitosis cell division did not occur, multicellular organisms would not have been possible. Then also it helps to maintain the proper size of the cell. Then it due to equational division, because of the number of chromosomes equally gets distributed among the daughter cells, the number of chromosomes remain equal and constant. Mitosis cell division is also the method of multiplication in certain unicellular organisms. Then another significance is that whenever there is a wear and tear in our body tissues, that means our body tissues gets damaged, mitosis cell division immediately, that means the cells immediately divide by mitosis cell division and thus restore that damage. Then if our body undergoes any sort of injury, the cells surrounding the injury divides themselves and thus heal the wounds. Okay, our next topic is tumor and cancer. All of you are well aware of the name of these two very serious diseases, that are tumor and cancer. Tumor and cancer are the product of uncontrolled mitosis cell division. So from the previous stages, you have got to know that mitosis cell division is a controlled process, and through this process, from one cell, two cells are produced, and from two cells, four cells are produced. This control of mitosis cell division is accomplished by a number of proteins, but sometimes due to some reasons the control of mitosis cell division gets hampered, and as a result uncontrolled mitosis cell division occurs, resulting in tumor and cancer. The reason behind this could be various types of germs, chemicals or radioactivity, or it can be virus. There is a virus named papillomavirus. This virus contains two genes. These are E6 and E7. These two genes, if enters our body, forms a chemical substance. This chemical substance can displace the two protein molecules which control cell division and thus result in tumor and cancer. So here is a figure explaining how cancer and tumor cells are produced. So these are the normal cells and they divide normally and in a controlled manner, but when the control is displaced, look at how a aggregation of cells are made. This is how a tumor or cancer is formed. Next, we are going to discuss about the meiosis cell division. Meiosis cell division is also known as reductional division. In case of higher organisms, meiosis cell division usually takes place before the sexual reproduction, that means during the production of gametes. And in case of lower organisms, meiosis cell division takes place after the formation of zygote, that means after the sexual reproduction. In meiosis cell division, the chromosomes divide once, but nucleus divide twice, and thus forms four daughter cells, each containing half number of chromosomes in comparison to their mother cells. Meiosis cell division plays immense significance in keeping the chromosome number constant in living organisms from generation after generation. So here is a figure explaining the concept of meiosis cell division. First, you need to know that meiosis cell division is actually a combination of two consecutive divisions. The first division is known as meiosis 1 and the second division is known as meiosis 2. In the first division, the chromosomes do not divide, and therefore from a cell two cells are produced which contain half of number of chromosomes than that of their mother cell. That means the meiosis 1 is actually the reductional division. So you can see that this primordial germinal cell containing diploid number of chromosomes, that means here you can see that 4 chromosomes are present and it became half by meiosis 1. The meiosis 2 is just like a regular mitosis cell division. That means from one cell and identical two cells will be produced. So in case of meiosis 2, from two cells a total of 4 cells are produced, each containing half number of chromosomes in comparison to their mother cell. So meiosis cell division is inevitable and plays immense significance in living organisms. Here is an example of how it does so. We are saying that meiosis cell division is responsible for keeping the chromosome number constant from generation to generation. But how does it do it? So, in case of sexual reproduction, we know that from primordial germinal cell or somatic reproductive cells, 2 individual gametes will be produced. That means 1 male gamete or sperm and 1 female gamete or ovum will be produced, which will then fertilize or fuse together to form a zygote, which is the first somatic cell of a living organism. So here we can see that the somatic reproductive cell contains a diploid number of chromosomes. That means a full set of chromosomes. If meiosis had not occurred during the formation of gametes from these 2 somatic reproductive cells, then the gametes would contain diploid number of chromosomes, and thus these 2 gametes after fusion would result in the formation of a zygote containing the double number of chromosomes than its mother cell. And if it had continued from generation to generation, then in each generation the number of chromosomes would get double, and this would have been a serious issue, and the chromosome number would not have been kept constant. So here is why meiosis is so important, as during the formation of these gametes meiosis occurs, the number of chromosomes becomes half, and as a result these 2 haploid gametes fuse together and unite together during sexual fertilization and forms a zygote that contains exact same number of chromosomes like its mother organism. Then again, keeping the chromosome number constant, meiosis cell division also maintains the stability of species. Another important significance of meiosis cell division is that during meiosis division, a phenomenon named crossing over, which is actually the exchange of chromosomal pairs, occur, and this crossing over results in bringing about significant variation among the offsprings. New characteristics, that means due to crossing over, new characteristics emerge, and sometimes these characteristics can ensure the survival possibility of a vulnerable species. Okay, so that's all from this chapter you had to know. Here I have collected some of the important questions that you can practice from this chapter. These are the stages of mitosis cell division. The figure and the identifying features of this mitosis cell division, the stages of mitosis cell division, is very important. You need to practice it as much as you can so that you can identify a stage without even a moment of hesitation. Later you need to practice the significance of mitosis and meiosis cell division. These 2 division plays immense role in the life of a living organism. So you need to know them, all of them, how they do so. Then you need to know how you can compare between these 2 cell division. That means the differences, the comparison between mitosis and meiosis cell division. Okay, so that's all for today. If you have any questions or queries, do not hesitate to leave me a comment or let me know. So stay well, then stay safe. Good luck.